Dr. Sam Raney is a thought leader in topical and transdermal drug products with over 30 years of experience in skin research. Dr. Raney has held several senior management positions in industry and is a member of many expert committees and panels of FESP. At FDA, he is the Associate Director of Science in the FDA's Office of Research and Standards and serves as a Chief Scientific Advisor for topical product bioequivalent issues at FDA's Office of Generic Drugs. Dr. Raney.
Thank you so much, Dr. Thakur. And thank you to Dr. Vinod Shah for a very valuable perspective on the history of IVRT. It really is a privilege for me to be here today to be able to tell you a little bit about what I've learned through the last several decades in my experience with IVRT studies, to talk about the fundamentals, both the scientific and in particular the practical considerations. So I will offer the disclaimer that this presentation reflects my own views and should not be construed to represent FDA's views or policies. So I think the starting point is to focus on the fact that the context of what I'll be speaking about will inherently talk about the IVRT in the context of the vertical diffusion cell. But I want to mention that there are different apparatus that are also utilized for developing IVRT methods, and these are described in chapter 1724 of the U.S. Pharmacopoeia. But by far the most commonly used one is the vertical diffusion cell that you see on the screen here. And the basic concept is that the dosage form is applied on top of a membrane, and through that it releases drug into a receptor solution, and by sampling that at different points in time, you're able to assess the rate of release. Now, the components of this are, of course, the dosage form that one would be working with, and typically you need to customize your IVRT method for that dosage form. And the customization, aside from the apparatus that you choose, which can often just routinely be the vertical diffusion cell or the diffusion cell you're used to working with, it's the membrane and the receptor solution, as well as other elements of the sampling that end up being key components of the method or the time points. So let me begin by saying that there is a guidance for industry on this because its use is quite well established for scale up and post-approval changes. But I will also be referring mostly to chapter 1724 because in the context of this workshop, it's not so much the scale up and post-approval changes in the SUPAC, but it's really the context of use for comparing any test and reference product, and particularly in the case of supporting a demonstration of bioequivalence. Now, in terms of 1724, it describes diffusion cell apparatus models. It provides some technical methodology for the use of each model, and it provides recommendations and guidelines for the test methodology. This became official in 2013, and it's currently under revision. So you'll be hearing in one of the sessions coming up today a lot more about what's happening with our revision of chapter 1724 at the moment. In terms of some of the key features of the in vitro release test, I want you to keep in mind that the IVRT membrane is typically a synthetic filter membrane, and that the receptor solution or medium typically contains alcohol or some kind of organic component. And this is because many topical products contain hydrophobic drugs, and you need to have adequate solubility for that beyond what you might just get in an aqueous receptor solution. Now, the IVRT methodology itself can be arbitrary somewhat and is quite product specific, but the dose itself is intended to be quite a thick dose, a pseudo-infinite dose. And the reason for this is because you don't want a small dose that is rapidly depleted. Instead, you want steady state kinetics. I'll talk more about that as well as we go on. Now, the dose is occluded again to prevent metamorphosis and drying, because you don't want that dosage form changing, whether it's that it's changing because the dose is being depleted or because of other reasons, because again, you want to support steady state kinetics here. And in fact, the primary result you're looking for is a steady state release rate, which is manifested as a slope. Now, drug is released into the receptor solution in fairly large quantities, typically in the microgram per mil range. And the dosage form and the receptor solution are actually in contact. They communicate through that membrane. Now, validated IVRT methods can exhibit really quite good reproducibility, and this is one of the great advantages of the test. But there's no expectation of an in vitro, in vivo correlation, and I think that's important to understand. But this release rate can be very sensitive, and here's the value, can be sensitive to physicochemical or structural differences between the test and the reference product. And by finding the release rates to be equivalent for comparably formulated products, it mitigates the risk that there may be these physicochemical or structural differences that could alter the product performance in a clinical setting. Now, I want to contrast this very briefly to the in vitro permeation test that we talked about yesterday. The design of the in vitro permeation test and the principles for that are fundamentally different. They're very, very different. This is looking at a finite dose. You're looking at a dynamically changing rate, not a steady state release rate. You're looking at very different fundamental principles for the test. And I won't go through each of these in detail, but I want to really highlight that these are completely different tests, even though they use, in many cases, the same diffusion cell apparatus. And it's very important to understand the differences. Now, the data that you get from an IVRT, an in vitro release test, measures the steady state rate of drug release. So here you have the cumulative amount permeated over time, actually mapped to the square root of time. And this slope, this rate, is that release rate that you're looking for. By contrast, the IVPT, the in vitro permeation test through human skin, measures the non-steady state, constantly changing flux rate over time of skin permeation for different formulations. In terms of method development, because I want to jump straight into that, the suitable method parameters as a starting point can often use the same VDC apparatus and dose amount and stirring rate, and even the sampling amounts or sampling schedules of, let's say, 1, 2, 3, 4, 5, 6 hours that you might routinely use for other methods. But what is particularly interesting to focus on is the receptor solution and the membrane, because these are very method specific and data dependent. And this is where I'm going to begin most of what I'll talk about this morning. Now, in terms of not only the considerations for the parameters, but in terms of what are you looking for to determine whether you've developed a suitable IVRT and that has good performance, the elements that you're looking at are the linearity of that release rate, the reproducibility of it, its ability to be sensitive and specific, and the solubility of the API in that receptor solution, as well as the inertness of the API binding to the membrane. There are actually quite a few additional considerations. We'll cover all of them in the morning here. Now, very briefly, there is one theoretical consideration that I want to touch on. Mostly this is going to cover practical issues, but there's an impact of this theoretical consideration. So there was a lot of excellent work done in terms of the foundation theory by Takeru Higuchi and William Higuchi. They were brothers. And William Higuchi, or Bill Higuchi, in 1962 confirmed the predictions of his older brother by fitting the solutions with empirical data that had been reported by Patel, Banker, and Decay. The interesting part here is that he further simplified the equations with square root approximations that end up being quite convenient for us today. And I want to focus in on this equation a little bit. Here, R is the percent of drug released at time T per unit of area of application with a thickness of H. We're going to come back to this equation in just a moment, but one of the reasons I highlight this article is because one of the things in terms of the steady state release kinetics is that he assumed that this percent of drug released throughout the conditions being considered were probably, in the range. It was safe to assume that you'd have steady state conditions if you were below 30% release rate. And I don't know if this is the source of what has since, I think, become maybe misinterpreted as the fact that there's something magical that happens at 30%, and that if your amount of drug, your dose depletion exceeds 30%, that somehow the IVRT is not valid anymore. In my opinion, this does not mean that IVRTs with a greater than 30% dose depletion are invalid, because an IVRT may well be able to sustain steady state kinetics at greater than 30% dose depletion. And I mention this because we've seen IVRT methods where they look excellent, but that, let's say after 3 hours, the dose depletion might be above 30%, and people truncate them because of a concern that they violated some scientific principle of the test. And so that's why I wanted to clarify that this is not a violation to go beyond 30%, that you just have to look at the data and make sure that you still have steady state kinetics. Now, in terms of that equation to which I referred, if you plug this in so that you assume that D is equal to 1, what you can see here is that at different heights or thicknesses of the dosage form, let's say if you have a very relatively thin, low amount of drug product dosed on the membrane, so it's about 2.5 millimeters thick, that over the course of the IVRT method, you would be at 100% dose depletion by the time you got to 5 hours. Now, what you can do is increase the thickness of that dose. This is just based upon the theory even. And what this would mean is that you now only be at 55% dose depletion. If you increase it further, that you are now running the entire test at 28% dose depletion. And I want you to appreciate here that one of the things you can do if you are seeing significant dose depletion, especially if it's beginning to affect the linearity of your release, that you can increase that dose. And this is what we mean by a pseudo-infinite dose and the value of that pseudo-infinite dose. Let me also show you some data that as you increase the dose, that you can end up seeing differences in the release rate. Now, you can see here as the dose increases, while the release rate increases, your depletion of the dose actually decreases. And this can be a very valuable tool in your toolkit as someone who's developing IVRT methods in order to be able to develop suitable IVRT methods. Now, I'm going to begin by talking about the receptor solution and the membrane. And I often encourage that when doing method development, that the first phase is to evaluate suitable receptor solutions. And you can identify candidate solvents and solutions for that receptor solution by looking at organic or alcoholic solvents in which the API has good solubility and stability. Focus initially on solvents that are miscible with aqueous solutions. And one of the advantages here is that aqueous, or I would say organic or alcoholic solvents, can be volatile, and that can make sample handling a little bit challenging. So it often helps to be able to have a hydroalcoholic mixture. And so focus initially also on solvents that are well suited to sample analysis. Many of these include ACN, ethanol, methanol, IPA, and so forth. Also assess whether the API needs, if it's, let's say, a hydrophilic API and you're using an aqueous solution, does it need buffer salts or does it need pH control? And it's helpful to evaluate different ratios of mixtures for that aqueous and alcoholic or organic component. All other study parameters being kept consistent, you often want to evaluate a minimum of 3 different receptor solutions, each in triplicate, in triplicate diffusion cells through your method development phase. And it's really helpful to utilize a preliminarily developed HPLC analytical method that's going to be compatible with all 3 matrices there. And then you can also begin to identify any, if like matrix effects and things like that. Ultimately, you also want to evaluate the solubility of the API in those receptor solutions. So empirically determine the solubility, because sometimes you may find that ends up being a deal breaker. You just don't have enough solubility relative to the sample concentrations you're receiving. When you've done this, so that's how you approach the method development in terms of the receptor solution. When you're looking now, you've got the data for 3 different receptor solutions. How do you pick one? Well, you want to select appropriate receptor solution and test parameters based upon looking at whether your release curve has some top-end drop-off, which means that you're losing steady state kinetics at the top end at the later phases, or if there's a bottom end lag, then that suggests that you are taking longer to get into that steady state region. And in either of these scenarios, you can either remove the last sampling time point or the first sampling time point and adjust your sampling schedule so that you really are in the most linear steady state kinetics region. Now, one of the challenges is sometimes you can have curvature and it still matches or it meets a R-squared criterion of 0.9, because that's incredibly generous. And what we've learned is that an R-squared value is not a very sensitive discriminator of linear versus non-linear behavior. And so that, while that's a guide, I really encourage looking, at very high R squared values, certainly 0.97 or higher. Don't just think that something is acceptable if it has curvature. If you can visually see curvature, try to adjust your sampling schedule. Also look at the percent CV for the linearity, look at the magnitude of release, look at something that preferably has a higher release rate. Look at the CV for your slope. And if you have a percent CV that's getting above 10% for your slope, in other words, the slope that you're getting across your different diffusion cells is variable, and variable with a CV of greater than 10%. This may be a signal that you could have trouble in the final developed method passing at a stage 1. So I encourage trying to keep that percent CV much lower if you can, and you typically can. You also want to prefer higher ratios of excess solubility capacity. You want to have as much excess solubility for that API in that receptor solution as possible, especially relative to the concentrations that you're seeing in your samples. And to consider whether the extent of API depletion is impacting your steady state kinetics. You also want to consider the matrix compatibility with the HPLC sample analysis method. Now, these are a lot of criteria, so you might want to consider some objective criteria to weight these so that you have a predetermined algorithm for how you weight these considerations so you can make some objective justifications for how you select this. And here's an example of evaluation of 3 different receptor solutions. And you can see that this one has a very linear release rate and relatively precise, but it's relatively low release rate. Here's a higher release rate, but you've got some curvature. This one has more variability in it. So when you're looking at these and you want to pick one, it's helpful to have some predetermined criteria, objective criteria to utilize. The next phase is the membrane evaluation. And part of the reason that I recommend doing the receptor solution first is because all of this involves doing analysis of your samples. And if you can lock down your matrix, you know what your receptor solution is first, that you know what your matrix is, and you can really start to fine-tune your HPLC analytical method, even for this next phase of membrane evaluation. So you'll use the one receptor solution that you used in the previous phase, that you selected from the previous phase, and you'll move that forward and now evaluate multiple membranes. In the previous phase, you would have just selected an arbitrary membrane, but now you're going to look at some additional ones. And some of the things that you can look for in candidates is the pore size. So a very common one is a 0.45 micron pore size in filter membranes. But something in the range of 0.2 to 0.8 is often suitable. And when this says pore size, I want you to understand that this is not as if there's these perfect pores or circles in the membrane that are exactly this size. That the membrane's going to look like this, and the dosage form, because of its semi-solid properties, is supported above the membrane, but the receptor solution, permeates through this, and so it actually communicates directly with the dosage form. So consider the pore size relative to the viscosity of the formulation. If you have an ointment, thick, you might want to have slightly larger pore sizes. If you have something that is more runny, you might want to have, something that has a relatively lower pore size. You also want to be considerate of the potential for the receptor solution itself to back diffuse into your dosage form. You definitely don't want that because it's changing your dosage form, and that's going to affect the steady state conditions. You typically want to have inertness characteristics of the material that you use so that it's not binding your drug. And you do want to consider whether it's a hydrophobic versus a hydrophilic material in the membrane so that you don't, again, encourage binding with your API. The chemical compatibility of the material with the receptor solution is also important because there are some organic solvents that may degrade the certain membranes. And the other thing to consider is this a membrane that is consistently commercially available? Because if you invest a tremendous amount in developing a method using some exotic membrane that 2 years from now is not going to be on the market, or that you can't supply, the supplier has no commitment to preserving that product line. Now you've got to go and completely redevelop your method. So one of the advantages with a lot of these filter membranes that some examples are below here is that they have consistent commercial availability over time, and that really helps. All the other study parameters being kept consistent, I encourage to evaluate a minimum of 3 different membranes, each in triplicate, and also in triplicate evaluate the binding of the APIs to each membrane by incubating it in that selected receptor solution for 6 hours at 32 degrees C. I'll talk more about this later. And then in terms of selecting an appropriate membrane once you have the data, you use criteria that are similar to what we talked about before for the receptor solution. Similar IVRT performance criteria as for the receptor solution evaluation, and see if you can identify a cutoff for accepting what your inertness test represents. Typically, this is going to be a solution where you have drug at a concentration relevant to the concentrations you're seeing in your IVRT method. And when you evaluate it at the end of 6 hours with the membrane incubated in it, you're assessing, do I still have 100% of what I had in solution, or have I begun to lose some because it's binding to the membrane? You might also lose some for other reasons. We'll talk about that later. But 100% plus or minus 5% recovery is a good target. Also consider the membrane compatibility, that it doesn't leach out anything that could be an issue in your HPLC sample analysis method, and prefer to avoid membranes where you're seeing any back diffusion. So again, consider some objective criteria to weight these factors when looking at the data, and then document that data evaluation so that you can justify how you eventually selected your membrane. I guess here's an example for different membranes. And again, you sometimes have to balance the criteria in terms of the performance of the release rate. You typically don't want to see high variability, for example. Now, it's optional whether you do this in method development or not, but one of the things that is nice to do is to get an early read on whether you have, with the receptor solution you've selected and the membrane you've selected and the time points you've selected, whether you actually have a discriminating IVRT method that can provide the sensitivity, selectivity, and specificity you're going to need. And so what you can do here, and I won't cover this in too much detail, is but to get an early read by looking at, let's say your product is a 5% product, to look at plus or minus 50% in strength, and to be able to look at how it discriminates the release rate from higher and lower strength products during method development itself, because that can give you that early read if you have a product, an IVRT method that is just not discriminating off the bat, that prevents you from then going into the entire validation process. This is an example of the kind of data you'd be looking at, where you've got your nominal reference strength product and then a lower strength, lower by 50% nominal strength. And so this is a 50% strength, 100% strength, 150% strength. You want to be able to see that it nicely discriminates the release rates even during method development. These release rates can then be plotted for those 3 different strengths, these 3 different release rates, to get a sense of whether you have relatively proportional release. And this tells you an early read on your specificity. Now, at this point, you're still not necessarily, you haven't validated your HPLC method. So you'll have to do this during validation with a validated analytical method. But one thing I want to highlight here is that you can see that this is not perfectly proportional. And again, the R-squared value is pretty high. I mean, over 0.95. Now, there may be reasons why you would not expect perfect proportionality here, because in the process of changing the product to put in a 50% more drug or 50% less drug, that there may be changes in the product that can impact the release rate. So this may not be perfectly proportional. So it may not be appropriate to expect 0.99 in this proportionality, but I think something above 0.95 is a good target here. 0.90 may be too generous in this instance. Okay, so final method development considerations to recapitulate. A single lot of reference product, or sometimes test product, is typically used throughout the method development study. The IVRT performance criteria for the selection of the receptor solution, of the membrane, or the sampling schedule is typically objective and predefined. The IVRT parameter selection criteria and the relative weighting of those factors we talked about are typically objective and predefined, with some allowance for justifications based upon the data that you're seeing. The objective analysis of the results are documented to support how you arrived at the selection of the method parameters, and the justifications are documented for the selection of receptor solutions and membranes for evaluation, as well as for the final developed method. Now, that final IVRT method with all the relevant parameters that define the method are then locked down, finalizing this is our method, and then you proceed into the validation step. Now, before we proceed into validation, that final HPLC method is also finalized so that it proceeds into validation, so that moving forward, you're now going to be using a validated IV HPLC method. So how does this look? Well, one of the things is you begin with having a analytical method that's going to be, so this is an overview of the method development process. You want to have some analytical method ready that's going to be generally compatible with the matrices of your different receptor solutions that you're going to evaluate in phase 1. And you do, comparison of 3 cells each of these receptor solutions, and then some solubility assessment for your drug. In phase 2, you would do 3 cells each of the 3 membranes, as well as some membrane binding. And then optionally, you might look at the comparison of the reference product as well as the low and high products. And this gives you a preliminary assessment of reproducibility, because you're going to be using the same receptor solution and membrane as you've already assessed for one of the conditions here. And now you're getting a preliminary assessment here. Now, this is just one way to approach it. There are ways to actually combine these and streamline things as well. So that's method development. We then proceed into the validation. Now, IVRT method validation has a lot of components to it. One of it is the apparatus qualification. Another is just making sure that your laboratory and your technicians are trained and able to perform the test. A key piece is the HPLC method validation. And then, independent of all of these, and certainly independent of the HPLC method validation, is the IVRT method validation. And here, you're evaluating several performance aspects of the IVRT method itself. And this lists are not all inclusive, but a lot of this information comes fundamentally from some precision accuracy type runs that you do on different days to show the reproducibility and precision of your method. And this also helps to define the linearity and range. And then another set that you do with lower and higher strength formulations. And from this, you get sensitivity, specificity, and selectivity. And then there's some additional things you'll do. So we'll go through these in one by one now. In terms of the apparatus qualification, USP 1724 has some good information about this. And it specifies that the diameters of the orifices of the donor chamber and the receptor compartment chamber, which define the dosage delivery surface area, should be sized within a tolerance of about plus or minus 5% to the specified nominal diameter. And there's some more information here about the diameter of donor receptor chambers. Can vary, but they should never be, one should never be smaller than the other, and that you should have good alignment between them. You should have consistent manufacturing, uniform height and geometry, and that the cells should have the same nominal value for the receptor volume, but that the true volume really should be measured for each individual cell, because the manufacturing has gotten very good in recent years, but if these are hand blown, the volume can be different, and that can affect your calculations. One of the other things that is mentioned in USP 1724 is that analysts should determine that the test temperature and stirring rate are within the specified requirements, and that you can actually run an IVRT with it, that you can do a performance verification test. And I'll talk more about this in just a moment. Also, equipment manufacturers may provide some supporting documentation like certificates of conformance that, where they themselves have measured the volume of each individual diffusion cell that are serialized and numbered. And that there may be guidance for installation qualification, operational qualification, and performance qualification, the IQ, PQ of these IVRT apparatus and some of the accessories for some of the more sophisticated and systems with a lot of automation with them. There may be also recommended schedules from the manufacturer for maintenance and requalification. One of the things we hear from manufacturers is dismay about how people maintain and how much maintenance they do to maintain their apparatus in good working condition. So what does this look like? So for example, when we're saying, determine the test temperature are within specified ranges or the stirring rate. So I'm going to talk a little bit about an example of what we had done with some diffusion cells, some vertical diffusion cells that we had. And we had different models of them. We had ones that were nominally 1 centimeter squared area that had a flat ground flange. And so we actually put a thermocouple probe under the membrane and then ran an IVRT for 6 hours and looked at how well the system was able to maintain temperature, and did the same thing for the 2 centimeter with the flat ground, the 1 centimeter with the O-ring, and a 2 centimeter with the O-ring. And here's some of the summary data for that. So this is an example of the kind of qualifications that can be done at the outset. And then once your apparatus are qualified, then from that point forward, you've got your qualification documentation. You don't have to redo it every time you develop an IVRT method with that. Here's a more broader set of criteria where, for example, I also talked about the stirring rate. So we would look at the actual magnetic impeller as well as the stir bars spinning in our different size diffusion cells. The nominal target was 600 RPM plus or minus 10%. In fact, the actual measurement was very, very tight. These often use clock motors that are very precise with a very tight percent CV. I talked also about measuring the orifice diameters relative to the nominal. So there was a target specification, and it came in with pretty tight CV. Looking at the receptor volume, there were some nominal targets here, actuals and percent CV. And one of the key things here is the actual release, the drug release, when you run an IVRT method. And here, we really want to have at least a 0.9, but this is very generous, like I say. In reality, they all had, greater than 0.99, and all the cells passed all these criteria as part of the apparatus qualification. This is a performance verification test that I think is incredibly useful. It's in the USP. You can look up this reference. It's a specific membrane and receptor solution and dosage form. And it doesn't matter whether you get the exact release rate that another lab gets using their apparatus. What's more important is that you're comparing a test and a reference set of this cream, the product to itself, and you want to be able to show that you can reproduce a good method that has good linearity, and that the products are found to be equivalent, that it's found to be equivalent to itself. I think this PVT is very, very handy. We'll talk more about it through the course of the workshop. Okay, so now when you talk about method validation, and we're going to go through each of these components step by step, one of the key things, again, is that the HPLC method used for the IVRT validation is validated in a manner compatible with FDA or ICH guidelines. This is really critical. And this is what differentiates some of the things that you're going to be repeating in method validation in terms of looking at the receptor solution solubility, your membrane binding. Some of these things that you did during method development, you're now going to do them with a validated HPLC method. So the IVRT validation characterizes the following critical method parameters that are outlaid here. And I've talked a little bit already about the apparatus qualification. I'm going to go step by step through the rest of these now. So the membrane qualification involves an evaluation of membrane inertness. And it's evaluated for drug binding in the receptor solution that you've selected at a concentration relevant to the average terminal release concentration. So you don't want to have a concentration in the receptor solution that is wildly different than the concentrations that you're seeing during your actual IVRT method. If you have a ridiculously high concentration in there, then even though you'd have significant binding to the membrane, and then all the binding sites are sort of saturated, because you started off with such a high concentration, you wouldn't see much loss relative to that concentration. And you'd say, I don't really have significant membrane binding. But if you then come down to the level that you're really at in terms of the concentrations of your samples, you may say, well, actually, relative to that, I had much more than 5% binding. I had more like 40% binding. That's why it's really important that the concentration in the receptor solution that you do this at, the concentration for API, is relevant to the sample concentrations that you're seeing. These determinations are typically based upon 3 replicate membrane incubations for the IVRT during that 6-hour duration, typically at 32 degrees plus or minus 1 degree. And these 3 replicate control incubations are performed in parallel, also without membrane. So you want to have something there where you've got the incubation, also in triplicate, let's say, but this time you didn't put a membrane in there because at the end of the 6 hours, you may not be getting 100% recovery, not because it bound to the membrane, but because you may have, some kind of a stability issue. And so you want to be able to monitor for drug loss that is not associated with membrane binding, and then compare what's happening in your membrane incubations to that endpoint. Aliquots of these solutions are collected before and after the duration of the incubation so that you can assess any decrease of drug in the solution, which you then can associate with membrane binding because you've already deconvoluted that it's not due to other factors. The recovery of the drug in the solution with an inert membrane is ideally 100 plus or minus 5%. This is the recovery of 100%, basically, plus or minus 5%. In terms of the receptor solution qualification, the solubility of the drug in the IVRT receptor solution is evaluated in triplicate by attempting to dissolve the drug in the receptor solution, typically at a high concentration, like mg per mil or whatever is appropriate for your IVRT method, and then assaying for the soluble concentration of the drug, the kind of saturation solubility. This solubility really needs to be at minimum in excess of what you'd be seeing in your terminal sample, like your 6-hour sample, for that 150% strength of the product, because that's going to have a higher release rate. And you want to be able to capture that. So you need to have a solubility that exceeds that, and ideally 10-fold greater than the terminal, for example, 6-hour sample for the reference product itself, because these are the levels that you're going to be seeing in your pivotal study. In terms of the sampling schedule, before I even talk about the schedule, just the actual process of sampling and qualifying your ability to sample the IVRT method well, the accuracy and precision of receptor solution sampling, that sampling collection at each time point should be qualified. And the evidence to qualify a sampling procedure typically illustrates that the sampling technique can reliably collect a consistent volume of the sample from the well-mixed volume of the receptor compartment at each sampling event, and that no artifacts are likely to be created by the sampling technique, for example, due to carryover between samples if you have an automated system, or due to sampling from the sampling arm and not really from where it may be unmixed in there. Often what people will do is at a sampling time point, if it's a manual and a vertical diffusion cell, is they might actually take from the center and then expunge that into the arm and mix what's in the arm with the well-mixed center. There are some additional different designs that actually help to keep things well mixed as well. And that information should be included describing the apparatus manufacturer's specification. This information should be included in the application, describing anything that the apparatus manufacturer has specified in terms of the accuracy and precision of receptor solution sampling, if that's available. In terms of environmental control, it's good to control the environmental ambient laboratory temperature and humidity, but more important, monitor it and report it. It's usually feasible to control the temperature, for example, in a range of 21 or 25 degrees plus or minus 2. This is about the control more than anything. You are going to have an occluded closed system for your IVRT method. So this may not be that critical, but it is helpful to monitor this. And humidity can be difficult to control, but it's, again, monitor this, and it's valuable to be able to report that. In terms of the performance of the IVRT method, the linearity and range, the precision and the reproducibility, and the dose depletion, all of these can come out of your runs that are typically done, 3 runs that I showed you before, with 6 diffusion cells each are dosed with the same lot of reference products. So this is 1 run of the reference spot, that's 6 diffusion cells on day 1, on day 2, and on day 3. And you can include different operators, analysts for the IVRT if you want to evaluate some other elements of this. So for example, here's what the data would look like. Day 1, day 2, day 3. There's a lot of information that then you can pull out of this. You'll get the linearity and range. That linearity, the R-squared value of the release rate slope, is calculated across the range of sampling times, which corresponds to the IVRT study duration. That linearity is compared within and across all runs, and typically you want that R-squared value, I recommend, to be greater than 0.97. So, while historically we've talked about 0.9, that really is far too generous. And this is something that we've talked about among USP experts as well, that 0.97 is probably a much better target for determining, and this is still reasonably permissive as a target for your R-squared value. In terms of precision and reproducibility, you report that. You report the mean, standard deviation, and percent CV among the slopes, and that's calculated within and across all the runs. And the intra and inter-run CV is, ideally less than plus or minus 50, less than 15%, sorry, not plus minus. And the reason for this 15% is really that your analytical method itself may only have been validated with standards where you have a precision and accuracy within about 15%. So it may not be suitable to then say you want a tighter precision than your analytical method can give you here. And so that's really where that number comes from. And then dose depletion is calculated. So you do this in a very simple way. The average recovery of the released drug in the receptor solution is characterized in each diffusion cell as the accumulated amount of drug in the receptor solution over the IVRT study duration. So cumulatively, how much was released. And you express that as a percentage of the amount of drug that was applied on the membrane, and that mass balance distribution of the drug between the product dose and the receptor solution and the associated recovery, that dose depletion, are calculated and reported. And this can be helpful for you to monitor because it can help you also to sometimes troubleshoot if you're having issues with your method. But by the time you get to the method validation point, this is just a reporting criteria so that FDA can assess some of the aspects here. So you also want to determine sensitivity, specificity, and selectivity. This is really important. It might be one of the most important things. Reproducibility is really important too. These determinations of sensitivity, specificity, and selectivity are based upon a minimum of one run of 6 diffusion cells for each of the 3 strengths of the product. The reference product strength, and then 2 altered strengths. You're going to have a lower strength, that's at minus 50%, and a higher strength that's at plus 50%. Now, if you have a situation where you're not able to produce a product at plus 50% strength, then some allowance can be made. But typically you want to have the 50, 100, and 150% products. And what you're looking for here is that with sensitivity, a higher concentration product, a higher strength product, has a higher rate of drug release, and a lower strength product has a lower rate of drug release. If you can show that, then you have sensitivity. When you then plot those, if you have that relatively proportional, and this is relatively linear, then you've got specificity. So sensitivity is the ability to detect changes in the release rate as a function of drug concentration in the formulation. If the IVRT method consistently identifies higher or lower rates of release for test products with increased or decreased drug concentrations, respectively, relative to the reference strength product, that's also run in parallel on the same day, then the method is sensitive. Specificity is that proportionality, is the ability to monitor proportional changes in the release as a function of drug concentration in the formulation. And we typically report this as the linearity. And I think 0.90 is again too generous, that an R-squared value of greater than equal to or greater than 0.95 is really a much better guide, so that you can look at it and start to eliminate things that may not really be very helpful for you in the long run. One of the next things then is to look at the selectivity. So not sensitivity, not specificity, but selectivity, the ability to select that, selectively show that not only is the release rate higher or lower for the higher or lower strength products, but it's different enough, it's higher or lower enough that it's actually differentiated as being inequivalent. This is very important because if it's not, that means that even in your pivotal study, you may be showing different release rates for your test and reference product, but you may not have adequate sensitivity or really selectivity to be able to discriminate it as being inequivalent. All of those tests are done with products at different strengths, but your actual pivotal study is going to be comparing a test and reference product at the same strength. So it's very nice if you can also show supplemental selectivity where you have the reference product as well as 2 other products that are altered, but all are at the same strength, and to be able to show that you can actually discriminate inequivalent release rates for different products at the same strength, which is what your test and reference product in the pivotal study are going to be. So selectivity is the ability of the IVRT method to accurately identify inequivalent product performance between the reference product compared with test products that have been intentionally altered. Intentionally altered how well, but different strengths. These alterations include different drug concentrations, but they can also include different excipients, excipient concentrations or manufacturing processes. And the determination of inequivalence is done using the statistical approach in USP 1724. The ability of the IVRT method to accurately identify equivalent product performance of the reference product to itself across the different days and runs also supports the reproducibility and the selectivity of the IVRT method. Because if even your reproducibility runs are found to be inequivalent to each other, well, then it's not that your method is selective, it's just not reproducible. So this is why you also want to show that for your reproducibility runs that they are found to be equivalent to each other. And then supplemental selectivity is performed similarly with test formulations at the same strength of the reference project. And so the way that this is done and what you do to alter them can depend a little bit upon the purpose of the study. Robustness is then looked at by looking at other method parameters like the temperature control or the stirring rate, and to be able to show where your method is robust. And if it's not robust to a particular parameter, then, well, it's really important to control that parameter and not let it vary. So robustness determinations are based upon the release rate, that average slope, of a minimum of one run of 6 diffusion cells for whatever that variation is, that variation in, temperature or stirring speed or whatever it is, typically using the same lot of reference product. And an IVRT method can be considered robust to the variation if the average slope of that robustness run is within plus or minus 15% of the average slope of the accuracy, the PNA runs, the reproducibility runs that you've had. If not, then that parameter may be identified as a critical parameter for the IVRT method. You've got to control it. You can't vary that. And so there's a few things that you can look at during robustness testing. So how does this look kind of as an overview? Just here's an example of the IVRT method validation approach, where it begins with making sure that your HPLC analytical method is validated. Then using your validated analytical method, you want to make sure that your membrane binding characterization and your receptor solutions to solubility characterization are assessed with that validated analytical method. And then here's an example of what can happen on different days. So for example, and you can do this differently too. Day one, you might do one run, 6 cells with the reference product, and then compare that to the low strength and the high strength. One of the advantages of doing this early on is to be able to show that you've actually been able to validate your sensitivity, specificity, and selectivity. If not, then you may have to go back to the drawing board. On day two, you're doing a run with the reference product again. So you're beginning to get some reproducibility information here. And this will also tie into when you do this a third time, you'll have 3 runs. And you'll be able to get your linearity and other information from here. But here you can do the altered test products and so we get that supplemental selectivity. And then on day 3, with your reproducibility run, you can also look at some robustness. This is just examples. There are other things that you can also do with some of the robustness testing here. So moving from the validation now into the pivotal study, some of the pivotal study considerations are that it's going to be performed in a manner compatible with what's described in USP 1724, where a single lot of each, the designated reference and test products, are evaluated and compared in parallel. And the objective here is to determine whether the release rate of the test product is equivalent to that of the reference product. That rate of release of the API, which is measured in vitro, it provides information about the features of the product that may impact product performance. And it's understood that in vitro releases, as measured by this method, it does not correlate with clinical bioavailability or bioequivalence, not directly. However, what you end up with understanding is that if you have, what you can get away from this, what you can conclude, is that if you have an equivalent release rate for comparable formulations, it mitigates the risk that there may be differences in the physicochemical or the structural features of these products, because those differences can impact product performance. These differences you worry about, because they're sort of at the level of the arrangement of the matter. And so by doing an IVRT and showing that there is an equivalent release rate, a release rate under steady state conditions that are telling you something about the arrangement of matter in that dosage form, and that that the formulations are comparable, the release rate is comparable, it's likely that you can mitigate a lot of the potential failure modes that may be associated with concerns for whether there may be, underlying differences in the arrangement of matter. So this is one of the ways that it becomes quite valuable, not only in post-approval changes, but also in supporting a demonstration of bioequivalence. In terms of the study design and its procedures, one test and one reference product lot are each prepared for dosing to 6 replicate diffusion cells. Now, I'm talking about vertical diffusion cells typically, using a validated IVRT methodology. The diffusion cell membrane is equilibrated. Very important that the system is equilibrated first, and then you dose, and that you're able to maintain this temperature throughout the test. The dosing is performed in a alternating pattern, alternating between test and reference products, and then following dose application, receptor solution samples are collected at timed intervals, routinely over 6 hours, however specified in your IVRT method. The receptor solution samples are then stored according to a validated method and then analyzed for drug concentration. Sometimes they may be put on the run, HPLC run the same day. And then the rate of release of APIs. And I put this as a note that sometimes you might have a drug product where you have more than one drug in there. It may be possible to develop one IVRT method for which you can analyze the release of both, but it really depends. Sometimes you actually cannot do that. You just need to have separate IVRT methods, one for each drug. The in vitro release rates for the test and reference products are going to be compared using a Wilcoxon rank sum Mann-Whitney rank test. This is all described in USB 1724. So I won't go into the detail too much, but I will say that if that first set of 6 cells by 6 cells of test and reference doesn't fall within the limits of 75 to 133, there is a second stage that can be performed with an additional 12 diffusion cells for each test and reference product. So rather than doing a 6 by 6, you can do an 18 by 18, and then look at USB 1724, and it'll guide on how that analysis of the data is done from there. This is going to be very helpful when you're using it to support a demonstration of BE. And there are some considerations that are unique in the context of using this to support a demonstration of BE. And that includes controlled procedures for blinding of test and reference products and for unblinding, and for statistical randomization of the dosing scheme. Is it going to be AB, AB, BA, BA? The statistical randomization of the retention product tube selection, and then some of the archival procedures for the retention product. I want to talk a little bit about the retention products, actually. So it's important that for any study supporting a demonstration of bioequivalence, that there are some elements that are important. And this includes the handling and retention of product samples, the maintenance of study records, and the reporting of any observations or deviations like SOP deviations or protocol deviations. And I'll refer you back to this slide. These slides and this recording of this presentation will be available after the workshop. But do refer to these CFRs and to these guidances, and do keep in mind that even though this is an in vitro test, that these regulations, which are bioequivalence regulations, do apply. It's very important that that's clear. So then when you do the IVRT pivotal study, this is the kind of data that you might see, where you'd be looking at the lower limit. And remember, we're talking about the tolerance, the range here are 75 to 133. And in this case, we're looking at it passing because it falls in 100 to 109, which is within the range of 75 to 133. And this is going to be for a well-controlled study where percent CV may be in the range of 2% CV or maybe 5% CV. And one of the wonderful things about these IVRT methods is that they can be very well controlled. But it goes beyond the scientific principles to control these. You also need to do this within a well-controlled quality management system. So the components of this system should include some of the it should incorporate some of the relevant principles of just good practices for all well-controlled studies. And this includes documented procedures for study personnel identification, training, qualification, and their responsibilities, study management and their responsibilities, QC and QA processes and personnel and their responsibilities, the utilization of standard operating procedures, protocols and reports, maintenance and control of the environment, and qualification and calibration of the instruments and the computerized systems. Very important, good documentation practices, and maintaining suitable records that facilitate the reconstruction of study events, as well as archival of study records. So I hope that this overview has provided some insight into not only what is done during an IVRT method development, validation, and pivotal study, but how it is done, how exactly it's implemented, and what the scientific principles are of the test, as well as what the implications are for then how you implement it. I would like to say that I have so many people to thank, but I do want to especially acknowledge some colleagues at the U.S. Food and Drug Administration, Dr. Priyanka Ghosh, Dr. Tanaz Ramazanli, Dr. Markham Luke, Hiran Patel, Sam Haidar, and Ahmed Zidan. I've learned a lot from so many people, including these people, as well as people outside of FDA, from whom I've learned a great deal. Paul Lehman, Vinod Shah, Kellis Thakur, Frank Sinner, Izzy Canfer, Thomas France, and really so many others. I consider myself a student of IVRT and a lot of the characterization of topical dosage forms, and I look forward to a very instructive afternoon. Want to not specifically mentioned here are my colleagues on the USB expert panels and so many others whom I've learned a great deal from, and I think we will all be learning from today. So thank you so much, and I look forward to a very fruitful discussion.
Thank you, Dr. Raney, for a wonderful presentation.